The overall aim of this protocol is to measure BK-polyomavirus non-coding control region-driven transcriptional activity by measuring dual fluorescent cells via flow cytometry.
The BK-polyomavirus can cause severe pathologies in immunocompromised patients, especially in renal transplant recipients. However, since highly effective antivirals are currently not available, methods measuring the potential antiviral impact of novel compounds are required.
This method allows virologists to analyze the impact of potential antiviral agents affecting the BK-polyomavirus transcriptional activity driven by the non-coding control region. This assay further allows to study the influence of rearrangements on the promoter activity in comparison to archetypical non-coding control regions.
The advantage of studying the viral promoter activity with a dual fluorescence reporter is that early and late gene expression can be analyzed in a mutually independent manner. Furthermore, the difficult preparation of viral stocks and long-term infectious cell culture experiments are not necessary. Since only transfected and fluorescent cells are analyzed, also drugs reducing the cell viability and proliferation can be studied.
This protocol follows the guidelines of human research as approved by the Ethics Committee of the Medical Faculty of the University of Duisburg-Essen. The first step is to collect blood samples for the isolation of polyomavirus DNA. Collect at least 3 milliliters of blood in EDTA acquisition tubes. Centrifuge the sample at 2500 G for 15 minutes and pipette the plasma into a new tube. Prepare 40 microliters of proteinase K into a 1.5 milliliter